Okay guys, so I told you I would make you a video on the cell membrane. So we saw a little bit about the cell membrane when we talked about biomolecules, but for this lesson I'm going to just cover the cell membrane in its entirety and why we learned all of those important biomolecules and how they play into our cell membrane. So first off, what is the cell membrane? Well, it's sometimes referred to as the plasma membrane. The cell membrane and the plasma membrane, they're synonymous terms. So if you hear either one of them, they mean exactly the same thing. And we like to think about the cell membrane kind of like the boundary between the cell and its environment. So I give a picture of like the Great Wall of China, where it separates literally China from the rest of the world. And your cell membrane does the same thing for your cell. It separates what is the inside of your cell from everything else that's happening outside of the cell. So it's like a boundary for the cell and its environment. Now, I have a question. Does every cell have a cell membrane? Well, if you think back to our three types of cells, right, where we had animal cells, plant cells, and prokaryotic cells, you will recall that yes, every cell has a cell membrane. Of course, animal cells, that's the only thing that they have. They have a cell membrane. Plant cells have a cell membrane right underneath their cell walls. And then, of course, prokaryotic cells, well, they have three types of external protections. They have a capsule on the outside, they have a cell wall, which is the middle layer, and then the inner layer, we have the cell membrane. So they have three layers. But yes, every single cell will have a cell membrane. Now, cell membranes, we say they help to maintain homeostasis, but what exactly is homeostasis? So is it cell breathing, is it cells getting blood to them, cells maintaining internal conditions, or cells reproducing? So those of you guys who are watching this at home, give you a few minutes to think about it. Okay, cells are maintaining internal conditions. We say when cells maintain internal conditions, they are in homeostasis, right? So that's almost the direct definition of what homeostasis is, just maintenance of internal conditions. Now, how do the cells do this, though? How does the membrane help with maintaining homeostasis in these internal conditions? Well, the cell membrane is what allows nutrients to come into and out of the cell. So, for example, some of these nutrients will want to come in would be like an example of glucose. And if you remember, glucose is a monomer of carbohydrates, right? So it's a carb. Amino acids are the monomers of protein. And then, of course, lipids are our fats and stuff like that. And so the cell membrane, these things will be trying to come in and out all the time to kind of maintain a balance. And our membrane is going to be what determines what's going to come in and what's going to go out. So we say that our cell membrane, one of its functions is it is selectively permeable. Now, these are new words, but the concept we've known before. So let's just break it down. Selective simply means that you are extra choosy on something. You're very choosy about it. So I gave the example in class, like when we talk about relationships. So I know many of you guys have boyfriends or girlfriends or are looking to have a boyfriend or a girlfriend. And when you select, we'll just say you select a boyfriend and girlfriend, you kind of check them out and see if that's the one you want to be with, you're being extremely selective. Many of you guys, I don't really see just any person who walks down the hallway, you guys are just going to kind of pick them up. I very rarely see this. You're actually very selective when it comes to your choice in who you want to be with in a relationship. Well, your membrane is going to be the same way. It's extremely selective, meaning it will only allow certain things to pass through. And those certain things that it's allowing to pass through are very small, uncharged molecules, like you see in this picture over here where I have my mouse over it. So small and uncharged simply means like water could pass through the membrane by itself. Certain gases could pass through the membrane by itself, like oxygen, carbon dioxide, nitrogen, things like that. Various other things that can kind of pass through might be some glycerol or some ethanol, things like that. These guys could pass on through the membrane. However, the membrane is very selective, so it will only allow these things maybe to come on through these small uncharged molecules. But these larger ones, these larger polar molecules, like different ions that you see over here, like sodium ion and calcium ions and things like that, or glucose or amino acids, well, it's not going to let those guys pass through without some help. Okay? So that is where my second word comes in, that permeability. That simply means that they are able to permeate or able to come through the membrane. That's what that means. So just remember, small and uncharged molecules, they're the ones that can pass on through. But the large ones, well, they might be able to come through, but it's going to need some help. And so that leads me to the next function of the membrane, which we say is helping in the removal of waste. So yes, your cells need to remove waste. They excrete waste, of course not like you and I, but they do have to remove waste occasionally. And so they're going to do it via this process called exocytosis, which I'll talk about in my next video. But I just want you to know that one of the functions of the cell membrane is waste removal. Okay, let's talk about the structure of the membrane. What exactly are all the pieces that go into our cell membrane? Well, let's see if we remember what this picture looks like. When I had first showed you this picture, I called it a micelle, right? It's what happens when phospholipids are dropped into water, and they form this bubble-like structure that has water inside of it and water outside of it, and then they can form what we call this lipid bilayer. Now, it's called a bilayer because bi means two. So lipid bilayer is just meaning that it is multiple of these guys all kind of lined up. And let's just recall really quickly what these things are that I'm pointing to. Phospholipids. So most of you guys will recall, right, that phospholipids was one of the main categories that I said we had when we talked about our biomolecules and under the lipids category. Now, the actual structure, okay, we're going to look at here in a second. Its first component on its structure, we say it has a phosphate head. So this phosphate head, if you'll recall, is polar in nature, meaning that it is sometimes attracted. It's attracted to water, and when we say it's attracted to water, we mean it is hydrophilic, right? Water-loving. So it is what is going to be the components that make up the outside of the border of the membrane. So it's going to be all the little circles as you saw in the other picture on the outside. Because remember, water is on the outside of our membrane, so therefore our hydrophilic phosphate heads are what's going to be touching that water. Also, if you think about it this way, I said it was a lipid bilayer, meaning that there's got to be two sides. So the inside of the membrane will also have phosphate heads on it because remember we have a water base in the inside of our cell. So if you recall the jelly-like matrix inside of the cell, we say it is cytoplasm. And cytoplasm is actually consistent of mainly water, which is why it kind of has that like liquidy jelly look because it's got a lot of water in it. So we could say that that's how we say that the cell membrane has water inside and outside. It's because of that cytoplasm and all that liquid it has. So I give you a little bit of a structure of a phospholipid. Now, this is slightly different than when I talked to you about the last time. Originally, when I talked to you guys, I just said it had a phosphate head and a fatty acid chains. But I'm going to add one feature in there I want you to know, and that's glycerol. That's this guy in the center of my picture. Don't worry about choline right now. It's not important because for these purposes of biology pre-AP, you don't need to know the intricacies of choline. But the three parts I want you to be aware of are the phosphate head, the glycerol in between, or the glycerol backbone, and then what we call the fatty acid chains. So glycerol, if you'll recall, we also find in another lipid, more specifically our triglycerides that we talked about. Remember, they have a glycerol molecule. Well, we can also say that phospholipids also have a glycerol molecule. Now, our last component of our phospholipid are our two fatty acid tails. And I really want to emphasize the two part. So phospholipids only have two fatty acid chains. I remember on the test seeing many little squigglies, I guess, that you guys were representing as the fatty acid tails, which the squiggles were fine. It was just you had about 4 or 5 or 6 squiggles on the phosphate head. This is incorrect. That's not a phospholipid. That's what I would consider a jellyfish. So remember, when you're trying to draw a very gross overview of a phospholipid, you only add 2 fatty acid tails. Now, if you recall, fatty acids are nonpolar in nature, meaning that they're not going to really mix with water. They're going to be hydrophobic or water-hating. So always keep that in mind, okay? They're what's going to make up the inner portion of the membrane. So all of these little tails are going to kind of mix in with each other to try and hide from the water that's both inside of the cell and outside of the cell. Now, I show this picture here just to recap. You'll notice that my phosphate, that's kind of a gross overview over here in the corner, you'll notice that it looks like one of its legs or one of its fatty acid tails is straight and the other is bent or kinked. If you'll recall, fatty acids can be both saturated and unsaturated. So when you see this little kink in this one leg off to the right, I want you to think that this one is unsaturated, right? It's kinked up because it has double bonds. It's unsaturated. While the straight one is representing a saturated fatty acid, which has only single bonds. So here's just another structure. Look at the phospholipid. You could see it any of these ways. They could just be kind of like gross blobs that you kind of see on the left-hand side, where it just kind of shows you a picture of like little circles and sticks. You could get a little bit more complicated when we talk about image B. This is more of a molecular structure of it. You're not going to be responsible for drawing the molecular structure of a phospholipid. I just want you to kind of see how we're getting this shape from the way that the molecules are structured. And then part C is what we call kind of like a filled-in ball and stick model, where you can kind of see like all of the molecules are being represented here. But this structure looks very similar to like what I think about Gumby or something like that. If we were to kind of fill in all of picture B and put it in as like little circles and sticks and whatnot, this is what it would look like in image C. So let's talk about polar versus nonpolar. Now, if you recall, okay, we say polar molecules, they have both a positive and a negative end. They're charged, right? So one side is a little bit more positive than the other side. And this is because there's an unequal sharing of those electrons. So electrons are negative, all right? So if I have two atoms that are sharing an electron pair, okay? The atom that has the electrons closest to it is going to be the one that is going to be more negative. That's because if the electrons are negative and it's holding the electrons closer to it, it's going to naturally be more negative. It's going to take on more of that negative charge from those electrons. While on the other case, the atoms that don't have as much of the electrons, they're just going to start getting a little bit more positive because those negative electrons are not influencing them as much. So they're going to be more positive. An example of this would be like water. Water is a perfect example of a molecule that is polar. On the flip side, we can have something that is nonpolar. And that means that they don't have charged ends. They don't have positive and negative ends. This is because the electrons are being shared equally. They're right in the middle. So everything is nice and balanced, and there is no charge struggle between the two atoms. So we say they're nonpolar, and they have no charges on either side. A good example of something that's nonpolar would be like fats or lipids. So which of the above would I say is hydrophobic and hydrophilic? Well, I would say that hydrophilic molecules are polar in nature, while hydrophobic molecules are nonpolar in nature. So why are my phospholipids arranged tail to tail? Well, it deals with that polarity we just talked about. So water, I said, is inside of my cell and it's outside of my cell. So if I'm a phosphate group, okay, and I like water, I'm hydrophilic and I like water, I'm going to want to be around the water. So I'm going to be wanting to be found on the inside of the cell and on the outside of the cell, because that's where the water's hanging out. But if I'm a fatty acid tail and I'm hydrophobic and nonpolar, meaning I'm water hating, I don't want to be anywhere near water. So I want to kind of hide with myself. Okay, so then you'll see all of these tails. The tails will kind of turn in on each other. And so you'll have tails kind of fitting together like little puzzles, and they're just kind of hanging out in the middle, trying to be away from all of this water that's around them. So let's see kind of how this will look. We say what happens is when this phospholipids come together, I told you they make a phospholipid bilayer, right? A layer bi means two, so there's two layers of phospholipids, okay, that make up that cell membrane. And this is kind of what it looks like. If you remember what I said from the other slide, I said that the phosphate heads are all on the outside, which you can see them. They're on the outside of the cell, down here. And then they're on the inside of the cell too, over here. So both heads are going to be facing where the water's at. And all of those non-polar tails that are hydrophobic, they're going to kind of be hanging out here in the middle, kind of away from everything, away from all that water. They're going to be hiding in with themselves. And if you'll recall, these little yellow things we're going to get to in a second, why we have these little yellow things all throughout here. And this is just another picture. We say that phospholipids are arranged tail to tail, again, due to that water both inside and outside. So here's another picture, of course, that kind of shows where the hydrophobic environment is. Now, when I say something is hydrophilic, do I mean it has a chemical makeup that likes water? Or does it have a chemical makeup that does not like water? So you could pause the video right now if you want and kind of think about it and then answer it. But I'm just going to go on right on through, so if you don't pause, the correct answer is number 1. Hydrophilic molecules have chemical makeups that like to be around water. Remember, hydrophilic is water-loving. So here's another question, and of course you can pause if you want to take a minute before I say the answer. Where would you expect to find water in the cell membrane? So if this is my cell membrane, right here, do I expect to find water here on the outside, or do I expect to find it here in that inside? So the correct answer on this guy is going to be number 1. It's going to be on the outside, right? Because water is going to be outside of the membrane, and it's going to be inside of the membrane, outside of the cell and inside of the cell, but never inside of the membrane, because that's where all those fatty acids are hanging out that don't like water. Okay, so let's talk a little bit about the next structure we're going to find in our cell membrane, and that's, lo and behold, cholesterol. Now, you remember seeing cholesterol when we talked about lipids in biomolecules, right? Cholesterol, we talked about it in there. Well, it also has a job or a function in our cell membrane. And its job is to kind of help stabilize phospholipids and keep them from sticking together. So our fatty acid tails, when they're hanging out here in the center, okay, they have a tendency to kind of want to stick with each other. And we don't want that to happen, because we want our cell membrane to be kind of able to move around and do what it needs to do. So we have cholesterol that's just kind of wedged here in between some of these fatty acid tails all throughout the membrane, okay? And you can kind of see it in this picture, all those little yellow spots. It's wedged in there all around to just kind of help stabilize the membrane and to help those fatty acid tails from sticking together in there. Now the next structure we're going to find a bunch of things that we like to refer to as membrane proteins. So we have 5 membrane proteins in total, okay? And membrane proteins, well, these guys are what help regulate which molecules can enter and which molecules have to be left out of the cell. So I told you we have 5 of them, so we're going to talk about 5 types. Well, there's more, but we're going to talk about 5 types that we'll use for our class purposes. Okay, and let's go ahead and just get on started with the first one. And we call this guy a carrier protein. Now, carrier proteins, okay, they're what allow the substances or the waste materials to move through the cell. So they're they'll carry in larger molecules that could not necessarily pass through the membrane on its own. So if you have a little bit larger molecules that need to pass through and they can't get through all of those phospholipids, maybe because A, they're too big, or B, they're polar, or they're charged, or they're they just cannot pass through that non-polar tails. We use that carrier protein to bring it on in. And so this is kind of an example down here in this picture of how it works. So what'll happen is we'll see these particles, they'll come on in, as you see that little the little red circle, and when it reaches the kind of the mouth or the opening of that carrier protein, if the carrier protein feels like it is necessary to come on through, or if it fits its shape properly, it's going to rearrange its shape and open up its mouth, allow the particle to slip on through, and then close on the other end, as you see here, in order to prevent other molecules from kind of like rushing in all at once on that. So we call this a carrier protein because it carries in substances or materials that need to move through the cell. Now the next one, we call it a channel or a pore protein. I usually will refer to it as a channel protein. And these guys, okay, these are hydrophilic channels. So they allow we say lipid insoluble substances. Alright, and what I mean by lipid insoluble is I mean anything that is polar and loves water. So anything that's polar and hydrophilic will not typically pass through, okay, these fatty acid tails. Partly because these guys are nonpolar and hydrophobic. So they're going to prevent anything that's hydrophilic from coming on through. So we have what we call these channel proteins, okay, that can kind of span the entire length of the membrane and almost make a gap for our particles to just kind of come on through that need to come through that are polar in nature or hydrophilic. And so you can kind of see that from the second picture of the image, the animation I have in the middle, where you see all these particles kind of going through to the membrane because of this channel protein that's kind of opening a gap. So if I were to get really specific on that, I might see this picture down here in the corner might also kind of represent that same thing, but that's a little bit more graphic in nature. Alright, so the third type we're going to say are our glycoproteins. Now these glycoproteins, I sometimes refer to them as the love child of two different macromolecules that came together. So if you'll recall, glyco, let's just break the word in half. Glyco sounds a lot like something else we know, maybe glucose or glycogen. So what macromolecule is glucose or glycogen? Well, if you'll recall, glucose and glycogen, they belong to the family of carbohydrates, right? So, in essence, we have a carbohydrate that is attached to a protein. And we call this a glycoprotein. So if a carb and a protein were to come together and make a little love child, they would be glycoproteins. And this is kind of what it does. Its function, okay, is to help with cell-to-cell recognition. Or what we sometimes refer to as, sometimes cells need to recognize if the cell is, if its partner cell or an external cell is one of it, if it's actually from the body, or if it's a foreign cell or something, in order to kind of communicate with each other to see, are you a part of me or are you not? We need to have some of this cell-to-cell recognition that happens, and the glycoproteins are what aids in that. And so you can kind of see them in the corner. The proteins are what the portion that's wedged inside of the cell membrane. And then these glycoparts, alright, these carbs, you see, they kind of branch out on the outside. And this is where we kind of get that recognition happening. Because they kind of are used as like receptors that kind of are like, hey, let me kind of see, are you like me or are you not? So that's kind of the job of the glycoprotein. Alright, so let's talk about more types of proteins. Our fourth one, we're going to say, is our receptor protein. Now, the receptor protein, these guys are a little bit interesting. They're different from all of the other proteins in the sense that they don't actually carry anything into the cell, okay? They're more like the messengers of the cell. So, if you look at my picture, you'll see a receptor protein here. He kind of looks like a wrench or something. And what happens is this receptor protein, it has this site right here, this kind of mouth or this opening that is a very specific shape. Remember, proteins, they're very specific in shape, right? Form follows function. They have to be a certain shape in order to do a certain job. And in this case, our receptor protein, the shape of its mouth or the entrance part, okay, is what matters. Because when particles come along, if they are the right shape to fit in our receptor protein, they're going to go ahead and bind. And when they bind to that receptor protein, instead of coming into the membrane, instead of coming through into the cell, what happens is this receptor protein is then going to send a message. It's going to send a chemical message that's going to float into the nucleus of the cell and tell my cell that it needs to do something. So it's almost like it's sending it a new message that says, hey, you need to do this job, or you need to do that job. And so these receptor proteins are really important from that. Now what happens after this little particle, or like you see in my picture it says a ligand, you don't really need to know specifically that word, but we'll just call it a particle. What happens after my particle binds and it's all done? It's done, the message has been sent. Well, very simply that particle will just pop off, it will be released, and it will float back off on its merry way. And that receptor protein will kind of regain its original shape and be ready to take in any new particle that wants to send a message. And so remember, we call these guys receptor proteins. Now the last one we're going to talk about are enzymatic proteins. And if you remember when we talked about enzymes, enzymes are a type of protein. Okay, but they're a type of protein that's very special in the sense that they are what catalyze chemical reactions, at least biological chemical reactions. And so we'll say that enzymatic proteins are what are going to catalyze our specific reactions inside of the membrane. All right, so if we need any reactions to take place inside the membrane, these enzymatic proteins are going to be kind of studied all throughout the membrane to kind of aid in that catalysis. Alright, so let's just recap as a whole kind of some of these structures that we found in our membrane. Just so you know, when you're talking about the membrane, it is very important to realize that there is a very distinct nonpolar interior region. And that was the ones that I told you is all that fatty acid region, those fatty acid tails. And we'll say that these guys are true barriers. So they truly separate the cell from its surroundings. And this is important, okay, because there are many particles, like sugars and proteins and everything that may want to come through, but they cannot cross this zone unless they are brought in by like a protein carrier or something like that. Partly because they're being repelled by this nonpolar region, okay? So unless you're nonpolar or uncharged or small enough, you cannot cross that membrane. And this is important because this really helps the cell regulate what's coming in and out, because it just doesn't want anything to come in, because then it could fall under attack, or a microbe or a bacteria or something could attack it. So it has to be very specific in order to kind of maintain that homeostasis and to keep itself safe. Sometimes we also refer to the cell membrane as what we call the fluid mosaic model. Now, the reason it's called fluid is because the lipid bilayer, that cell membrane, it's not strong or firm like you would think of like a cell wall or a shell. It is actually very fluid and it's always in motion. It's kind of like a soap bubble. It's very fluid-like. It's called a mosaic, partly because if you've ever seen a mosaic, you know that it's a piece of art that's made up of lots of little pieces of maybe like ceramics or tiles or glass or whatever. It's got lots and lots of pieces kind of all over to make a picture. Well, the cell membrane, we learned it has cholesterol in it, it's got fatty phospholipids, it's got 5 different types of proteins in it, It's got all sorts of stuff that's all inside of this membrane. So it's kind of like a mosaic in that sense, where it has many different things all throughout it. Now, individual phospholipids, just to remind you on this one, they're arranged, okay, kind of side by side, and they kind of float within that bilayer, okay? And of course, just as a recap, cholesterol is what prevents our phospholipids from sticking together. So this is in essence all of what you'll need to know for our cell membrane. If you're doing your cell membrane worksheet, okay, which is attached to the back of your notes, just a little thing that you should know when you turn it in for homework, there are a few blanks that I have on there that I did not discuss in class that you won't be responsible for. So just so you know, these blanks are as followed. Part B is actually supposed to be a glycolipid. Okay, glycolipid. I didn't talk about that one. I only talked about glycoprotein. Okay, part H is actually what we call a peripheral protein. I didn't talk about the peripheral proteins, but just so you know, that's what it is. That's going to be letter H. And then letter J on your handout is going to be the cytoskeleton. Okay, so if you recall, all cells have a cytoskeleton that kind of keep its shape. Well, that's what's being represented in part J. So that's all for the cell membrane notes. If you have any questions, remember, come see me for tutorials. I'd be happy to sit down and help you.